Yes, yeah.
Great. All right. I'd also like to acknowledge that I'm meeting with you this evening on the land of the Gadigal people of the Eora Nation, and I also pay my respects to Aboriginal and Torres Strait Islander people who might be with us or listening, and recognise that the lands of Australia have always been places of teaching and learning and healing. So I'm going to talk about biobanking. And before I do that, I'll just give a little bit of background to my role. So I'm Director of Biobanking in New South Wales Health. And so I really have four components to my role. I'm here to provide leadership to New South Wales Health biobanking programs, particularly the New South Wales Health statewide biobank that I'll introduce to you. I'm here to drive collaborations to achieve New South Wales Health's biobanking objectives and improve services. I'm also here to carry out research, and I primarily do that through my role at the University of Sydney. And I'm also, another responsibility is to lead and advocate for biobanking workforce planning and skills development. So tonight I will give you an outline to what is human health biobanking before I talk a little bit about the New South Wales Health Statewide Biobank, where I've been for the past 6 years. And then towards the end, I'll talk a little bit about how biobanking could potentially impact the use of animal models in research. This is one of my favorite quotes in science because it can be thought about in different ways. It was, I think, quoted around the turn of the last century. And it was Henri Poincaré, who I think is a French mathematician, who said that science is built up of facts as is a house with stones, but a collection of facts is no more a science than a pile of stones is a house. So it is essentially this organization of knowledge that builds, I guess, direction within science. And we start with what's often referred to as basic science at the bottom, while I'll also talk about that as preclinical research. So a lot of this research is conducted in laboratories, and then some of the results of that research can be carried out in a more translational setting before we actually start testing new treatments through clinical research. And then again, a fraction of that research will be translated through to patients in the kinds of medicines that people will receive in a healthcare setting outside clinical trials. So it's a stepwise process, and we need organised and robust knowledge to allow us to progress through all of these different stages of research. Within I guess I've been a scientist for quite a long time, and I'm also fond of this particular diagram, which came from a paper published in 2017. It highlights how scientific productivity requires at least some emphasis on both quality and quantity. And there is an optimum that can be achieved between these two, to some extent, opposing forces. So if researchers are overly concerned about the quality of their work, that can actually hinder productivity because people become overly perfectionist. They don't finish things, they don't publish things. But then if they are focusing too much on quantity of output, such as quantity of publications, we can end up publishing more incorrect results because there's not enough attention paid to the detail that actually underpins the rigor of the work. So research is a balance, and I will talk a little bit about this later as well. Some of you will be aware of the fact that particularly in biomedicine, we've been talking about a research reproducibility crisis since about 2012. And in Nature conducted a survey of all scientists, asking them whether there was a reproducibility crisis in 2016. And about half of the people who replied said yes, they thought that it was a significant crisis. They thought that there was a significant problem. And this has been also, I guess, supported by studies of the extent to which particular results are reproducible. So preclinical research, this basic research that I was talking about earlier, is really the bedrock upon which everything else is built. But preclinical research has a number of features. It is expensive to carry out. It is often very technically difficult, and it tends to be very slow. So because of all these factors, it's actually quite difficult to reproduce preclinical research. We don't really have a culture of reproducibility, and we don't really support it. So lots of research is conducted and it's never reproduced. But when people do try and reproduce it, they often find that there are problems in doing so. Hence, this description of the reproducibility crisis. So biobanking, so that's just a bit of background to sort of some of the issues in biomedicine at the moment. So biobanking is a particular process that's designed to support biomedical research. Essentially, biobanking supplies, in the human context, it supplies samples that come from humans and their associated data for research. So biobanking has some strengths. It can enable relevant research because we are using samples that come directly from human participants. It can also enable the derivation of other kinds of models, such as cell lines, which are homogeneous cell populations that people grow in laboratories, or organoids, which are more organised collections of cells that grow in ways that they start to mimic organs as they occur in living organisms. So it is really the combination between the biospecimens and the data that's attached to them and the quality of both of those attributes that determine a lot of the research that can be undertaken with human biospecimens. So biobanking, by delivering these quality samples and data, can really support biomedical research. So at this point, I want to just highlight that there is a slight difference between biobanks and biobanking. So biobanking is the process, whereas a biobank is a facility where this happens. A biobank's a building, but biobanking is more than just the building, as I'll show you. So what the biobanks do is essentially they manage what's called the biospecimen life cycle. This term was first coined by Helen Moore et al. In 2011, so some time ago. So biobanking involves consenting patients or participants and then obtaining the sample and then possibly collecting that sample and transporting it somewhere so that it can then be processed. Then the sample can be stored, and then once it's needed for research, it can be shipped and then analysed by research. So this is what's been referred to as the biospecimen life cycle. So in part of this life cycle, we see samples and data going into the biobank, which is where they're stored. And then the other part, we have samples and data going back out to researchers. We can also have the return of samples and data to the biobank in some circumstances. So this can kind of be a circular pattern. So it's important to recognise that not all biobanking actually happens at the biobank. Some biobanks oversee the entire life cycle, including doing research on the samples that they store, but many other biobanks might only, for example, just do storage and shipping, or they might process, store and ship the samples. So there's a variety of different ways that biobanks can support biobanking. So for example, specimens and data can often be sourced externally to the biobank, and similarly, sometimes complex processing can be undertaken by research teams before those samples are given to the biobank. And of course, a lot of the time the analysis is done by research teams who are outside the biobank. So this overall process can involve different kind of research actors, if you like. So biobanks can store a variety of different types of biospecimens. We generally refer to the sample that's taken from the participant as a primary biospecimen, for example, blood or a tissue sample. And these kinds of samples are stored unfractionated in the biobank, so they can either be frozen whole or stored at a different temperature, but not in a fractionated state. We can also take primary specimens and then process some of those to produce what are called secondary specimens. So those secondary specimens can be things like cell lines, which I mentioned briefly. So these are homogeneous cell populations that can be grown generally indefinitely, but this growth takes place usually in cell culture dishes in laboratories, so in quite an artificial environment. We can also, from tissue, we can derive organoids that mimic certain attributes of tissues, such as, for example, gastrointestinal tissue. And we can also obtain samples that might have been grown in other organisms that can then be stored in the biobank as well. So essentially, biobanks can store a vast range of different kinds of samples. There are different models for biobanking. I won't go through these a lot because really this could take a fair bit of time. But essentially, classic biobanks are those that store samples that tend to be around a particular disease, and they're usually collected retrospectively. So people will have obtained a cohort of samples, and then they'll store those with the intent of supporting research on that particular condition. Prospective biobanking is where samples are decided to be collected for the future. So people might say, I want to create a particular collection of this, and then they go about very systematically creating that collection. Population-based biobanks, such as, for example, the UK Biobank, will have information and samples from very large numbers of people. So obviously these are great for population health studies, but they are expensive to establish and maintain. Archival biobanks are usually established in pathology departments in hospitals where samples have been collected for clinical care, but then can sometimes, under certain circumstances, be made available for research, and consent can be an issue here. And then finally, data-focused biobanks are those that really have largely converted the samples to data, and then they provide the data to researchers for different kinds of work. So yeah, there's different biobanking models, and I'd say probably in Australia we have most biobanks essentially operating through this classic model. We can also classify biobanks according to the type of researcher access policy that they allow. So open access biobanks are biobanks that allow different kinds of users to use their samples. And according to our research, these tend to be larger biobanks. They're formally constituted biobanks. They often have websites. They're often outward-facing organisations. Restricted access biobanks might only allow sample and data access to either a single research user or small numbers of users. And these biobanks also tend to be smaller biobanks themselves, although there's probably exception to this rule. So now I'm going to give you a bit of an introduction into the New South Wales Health Statewide Biobank as an example of a biobank. Because biobanking is actually not a very expensive activity to enter, and because there are, in countries like Australia where we have an organised health system, we have very ready biospecimen access directly through healthcare. So these two things can combine to create a lot of individual biobanks. And so what we have in Australia, unfortunately, is a very fragmented health biobanking sector. So this can have a number of consequences at a system level. A lot of these biobanks can be poorly visible, so people don't know about them. They can often be under-resourced because they're often started with minimal resources, and this can lead to underuse. This might not sound like a very big deal, but it is really a significant problem. If biobanks are underused, obviously we're losing research opportunities because biobanks are created to support research. Over time, this lack of use can lead to waste of resources, including the goodwill, which is often relied upon to establish biobanks in the first place. If biobanks are underutilised, they lose capacity for cost recovery. Many biobanks try and recover at least some of their costs, but they only recover their costs if they're actually being used. And very importantly, if biobanks are underutilised, they lose the capacity to actually learn and improve on their processes over time. So the statewide biobank was established partly to counteract the continued establishment of smaller biobanks. So as an organisation, it's supported by the Office of Health and Medical Research here in New South Wales. It's actually operated within our state pathology service, so that's the New South Wales Health Pathology organisation, and we're located in a local health district, so the Sydney Local Health District, which is in the centre of Sydney. So the biobank was built as a biobank, and it was opened in November 2017. So, that's a number of years ago now. Unlike many biobanks, it was purpose-built, and we have also achieved certification to the quality management standard of ISO 9001. The statewide biobank is largely built around a prospective collection model, meaning that the biobank helps researchers create collections for their own research use, although of course we do have some other kinds of collections stored at the biobank at the moment. And we aim to cost recover at least a proportion of our operating costs so that we can offer flexible services and improve services in the future. So this is a picture of the building that we're located in. We're located in the Professor Marie Bashir building, and we occupy almost the entire first floor of that building. So it's about a 650 square metre floor plan. The aims of the biobank were firstly to enable new biomedical research and also to help attract new national and international funding for research. We aim to partner with existing New South Wales health biobanks to improve the availability of samples for research and to also help with this quite difficult process of rationalising and centralising health biobanking infrastructure. The statewide biobank has a number of unique capacities compared with other smaller biobanks. Firstly, there is a very large storage capacity at the biobank of up to 3 million samples. The laboratory facilities are quite extensive and they're built around automated processing and storage, which is currently quite unique in New South Wales and also one of only a very small handful of biobanks in the country that are operating in this way. So through New South Wales Health Pathology, the statewide biobank can offer sample collection across the state and also linkage to health data sets through the Centre for Health Record Linkage, or CHERL. And a number of projects are supported by biospecimen collection grants that were previously offered by the Office of Health and Medical Research. So we have a lot of these things are not typical activities of other biobanks. So again, working at the state level, the statewide biobank has the capacity to support research at scale. So that's large project numbers, larger project scope, as well as complexity through a greater diversity of different types of research clients and a large range of research topics that are themselves often quite diverse. So the statewide biobank also aims to offer leadership in biobanking, and I'm just going to talk about one particular activity that's been happening at the statewide biobank for some years, and that's the New South Wales Biobank Certification Program and Biobank Register. So this program was established in 2016, and I was one of the people that actually brought this program from Canada to Australia. So it's a voluntary certification program that shares education and best practices in biobanks. And since 2016, we've created a biobank registry of over 50 biobanks that I think in terms of its scope is unique in Australia. There are a couple of other biobank registries now operating, but this is probably the one that's been running for the longest. So if you want to register for the program and access the modules, you can visit the website that's shown on the screen there. So if biobanks actually want to certify, they need to undertake a registration step, which involves completing an introductory education module. And then the certification process involves the different staff members of the biobank completing assigned education modules. And then there's some confirmation of biobank documentation, submission of a declaration of compliance, and then biobanks can be certified. So the program offers customised opportunities for biobank staff, but also for really anybody who wants to learn about biobanking. And I think when we established this in Australia, we conducted a study to actually determine how much it costs. And we found that participating in this certification scheme is quite affordable. So it's a good entry sort of level activity for biobanks that are wanting to improve the quality of their operations. So just to show that again, if you visit the website, you can learn more about the program, register and access some of the modules. So finally, I'm going to talk a little bit about how biobanking might impact the use of animal models in research. So coming back to this idea of the reproducibility crisis and the fact that research is, expensive, difficult, slow, and sometimes producing answers despite everybody's best efforts that don't reflect the disease that people are trying to study. So this slide shows, I guess, preclinical research without biobanks in a very simplistic way. So often researchers start carrying out experiments with cell lines because they're inexpensive, they're often available within research facilities, they require the kind of equipment that people often have on hand. But of course, cell lines have some limitations. Depending upon the disease under study, there can be limitations in the overall number of cell lines that are available, the degree to which these actually represent the disease of interest. Obviously, the culture conditions for growing cells in dishes is very different from the kinds of conditions that cells experience inside, a human body or another animal. And this can lead to issues around the generalizability of these results. You might obtain results that apply in the particular cell line models that you've tested, but then when you try to translate those results through to animals and even people, this can lead sometimes to results that don't replicate, or potentially results that do replicate but aren't actually relevant to human biology. So if we add in the capacity to study human biospecimens into this sort of pipeline, we might start doing some experiments in cell lines and then expand those results through a collection of human biospecimens and data from a biobank. So here we can have the possibility of potentially looking at greater numbers of samples from a greater number of people that are more representative of the overall disease as it occurs in human populations, which might increase the likelihood of actually having results that are more generalizable and translatable. So then if we start with better data that's coming from, for example, biobank samples, then what might that mean in terms of animal experimentation? It might mean that the design of those experiments is improved. It might mean that fewer animals need to be used, and that the use of those animals is more likely to lead to data that actually translate through to human participants. So if we think about, for example, we might be able to actually remove cell lines from this equation. We could start studying human biospecimens and then actually use organoid models instead of cell lines before carrying out any experiments in animals. I guess we have the same possibilities of maybe having models that are greater, that are more representative of the disease under study, that produce more generalizable results, and that'll have possibly a similar impact upon the use of animals in experimentation. Could we remove animals from this equation? I mean, that's certainly not something that I can say could always happen, because I'm not really an expert in terms of the use of animals in research, but there's certainly a lot of people that are very interested in this concept of using organoids and carrying out experiments in these kinds of systems to replace animals. But certainly what we're going to need if we want to do this is we need scaled investments, in model development and also in the biobanking infrastructure that will support those. We need to maintain a very firm focus upon quality and also relevance of models. And finally, work towards making sure that all of these kinds of resources are accessed more easily by more researchers. And I think in the meantime, what we can all do as scientists is focus on improving research quality at every stage of this pipeline, because it is a pipeline. So if we start with kind of data that aren't robust here, we have a very poor chance of translating our data through to people. And on the way, we can end up doing unnecessary experiments that can involve animals. So I think really focus on improving research quality is something that I think is it's certainly something that we can focus on achieving in the short term. So I'm just going to finish by acknowledging the members of the statewide biobank team. Certainly biobanking, I haven't talked at all about people, but actually biobanking is everything. Everything about biobanking is really achieved by human beings working in biobanks. And at the statewide biobank, we're very fortunate to have an excellent team. So I've listed all the current members of the statewide biobank team, as well as my research team at the University of Sydney. I'd like to thank you very much for your attention and very happy to answer any questions. Thank you.
Thanks so much, Jenny.
Thank you, Jenny. We might just with that